request Dr. Shishendu Mukherjee, Mission Director GSAI, for opening remarks and providing overview about Grand Challenges India initiative. Over to you, sir. Thank you.
Thank you, Dr. Richa, and good afternoon and namaskar from BIORAC, Department of Biotechnology and Grand Challenges India team, and Bill and Melinda Gates Foundation. We are happy to host this webinar to tell you all about our Grand Challenges call on the diagnostics for lymphatic filariasis, which is a major challenge for this part of the world, and it's a strong unmet need. Today, in this webinar, I have with me my colleague from the Gates Foundation as well, Dr. Bhupinder, my colleagues from BIORAC, Richa, Madhvi, and other colleagues who will take us through this journey of how this call has been designed, defined, and what are the unmet needs, and what are the key features of this call, and how you can target and take maximum benefit of this call to take it forward. At the end of this session, we will have a question-answer discussion. You can type your questions in the chat box as well. Following which, after this call, we are always available through our registered numbers as well as through our emails for any queries. So I'll first try to give you in another 5 to 7 minutes what is this program all about, what is Grand Challenges India actually, and how this call is designed and fits into the mandate of Grand Challenges India. So Grand Challenges India is a joint initiative between the Department of Biotechnology, Government of India, and the Bill and Melinda Gates Foundation, and is implemented through the Biotechnology Industry Research Assistance Council. And the main mandate of Grand Challenges India is to improve public health and beyond. Broadly, we work on a lot of thematic areas. The next slide, please. A lot of thematic areas, and as I said, it is a co-funding between DBT, the Bill and Melinda Gates Foundation Wellcome Trust, and it is housed at BIORAC. With our identified partners, we work in the public health need areas and try to target unmet need areas and take it to a conclusion so that we can come out with solutions to these unmet need areas and take it to a level either which can be de-risked to be implemented or can be converted into a policy for a bigger implementation program. Next slide. And we work in various areas of maternal and child health, agriculture, nutrition, infectious disease, wash and sanitation, medical technologies, vaccines, immunization data. And the idea of Grand Challenges is to work in very close assimilation and relationship with the national programs which are government support. We work very closely with the Swachh Bharat Abhiyan, National Nutrition Mission, Make in India program of the government, and Atmanirbhar Bharat initiatives. We work across the Women and Child Development Ministry, the Ministry of Health and Family Welfare, the Indian Council of Medical Research. We work across them and we try to synergize the program which our country needs. And all is this implemented by the Program Management Unit, which is housed at BIORAC. The next slide, please. So this is how the evolution of the Grand Challenge India took place. We started our journey in 2012. We are just completing our 10 years of existence. Our partnership with the Bill and Melinda Gates Foundation and Department of Biotechnology is completing 10 years on 18th of July. And during this 10 years of our journey, we worked on various initiatives which I just described, starting from agriculture nutrition to the wash and sanitation, to children's maternal and child health initiatives, to exploratory programs as well from the Grand Challenges Exploration Plot, where we literally farmed innovations and plucked the good innovation to take it to the next level. And very closely with BIORAC's program as well, we try to integrate our programs with them. We recently worked on the medical technology challenge as well, the vaccine challenge, the diagnostic program. So we have a very broad portfolio of 30 plus programs, and we work in the different areas, starting from basic science to policy to translational research to clinical trials, and we have a broad portfolio of programs in that space. Next slide, please. These are some of the important programs. I'm not going to describe that, but this is the portfolio of programs which Grand Challenges India runs. And all these programs are in close assimilation with the Government of India through Department of Biotechnology. We run those and cross-ministerial interaction with various other line ministries. It gets implemented. Next slide, please. So this is what the program we are all going to talk about, and you are all here to discuss about these programs today. This is the diagnostic call which we have announced on 21st of July and will, sorry, on the 7th of June, and it will close on 21st of July, sorry. And this is a very important call with strong unmet need and which our country faces. We would look forward to innovations to address diagnostics for lymphatic filariasis. And we would like all of you to take maximum advantage of this call to take your innovations to the next level. Next slide, please. So I think this is what I wanted to add to give you a flavor of Grand Challenges India. Now I invite my colleague from the Gates Foundation, Bill and Melinda Gates Foundation, and he leads the lymphatic filariasis program within the foundation at the country office. So I request my colleague Dr. Bhupendra Tripathi to enlighten us about this call and how we can take maximum advantage from this call. Over to you, Dr. Tripathi.
Thank you so much, Dr. Mukherjee. It's a pleasure to be with all of you for this session. So, yeah, let's start with the presentation. So, this is a program which is largely run by the National Vector-Borne Disease Control Program, and we are trying to engage with the Department of Biotechnology, BIOMARKERS team in specific, to figure out how we can support the program in figuring out new NF diagnostics for the country. Next, please. So in the presentation, we will take you through the introduction about what is LF, transmission signs symptoms, and the problem. Then what is the public health priority for the elimination? Then what are the steps which are already being laid down by WHO on elimination? Then role of diagnostics and why and where they are used. And we will also share about the WHO guidelines regarding the new diagnostics, and then the potential market in India and abroad, and then about the call which we have just heard. So next, please. So in terms of this disease, it is known as lymphatic filariasis. In common Indian parlance, we call it filaria or hathi pav or field pav. And this disease is a parasitic disease. Basically, there are parasites which are known as Bruchella bancrofti, Bruchella malai, and Bruchella timori. So these are small worms which are in the body, and they have specific geographical focus. In India, more than 95% areas we have Bruchella bancrofti as the specific parasite, and this parasite is spread by mosquitoes. Like a person having the disease, if a mosquito bites that individual, and then the same mosquito bites another individual, the disease is transmitted. Next, please. In terms of the life cycle, if we see, so this is definitely a disease which is mostly in tropical areas, and because it happens in the neglected population of the community, it is known as neglected tropical disease. And we are having around 20 diseases which are part of the NTDs, and this is one of them. So this disease is basically, and as I told you, spread by mosquitoes, and the life cycle is both in human beings as well as in mosquitoes. So if you see the picture on the left side, in this diagram, the adult worm basically is the human body, but the adult worm gives babies which are known as microfilarias. And these microfilarias are, having stage one, larva one stage, larva two, and larva three stages. Mosquito bites human beings and takes the larva stage one, and then the maturation happens inside mosquito, and the larva 3 is a vector which is basically infectious. So when larva 3 is injected by mosquitoes into a healthy individual, then that larva turns up into adult worm. So the male and female adult worms get developed, and they reside either in the lymphatic channel of the body or in the lymph node of the individual. And when they are alive, they are mostly mobile. Their mobility is visible. Even the lymph nodes, if we try to do ultrasound, we can find a very nice filarial dance of these worms. But over a period of time, like in 6 to 8 years, these adult worms die. When they die, then there is an antigen antibody reaction in that specific area, and that area becomes highly fibrosized, leading to swelling in the distal part of the body. So if you see the stages of lymphedema, we grade them into grade 1, 2, 3. And the grade 1 is basically swelling which is coming in the morning, and perhaps with some rest or elevation of your feet it goes away. So that is grade 1 lymphedema. But in the grade 2 lymphedema, the swelling persists, and when the swelling is accompanied by nodules and warts, that become grade 3, which is a severe form of lymphedema. And mind you, we do not have any clinical treatment, whether it's drugs or injections or operations, which will treat this infection. Next, please. So I will not go into the details, but I would just like to tell everyone here that this disease usually occurs in the childhood. Like the infection happens during the childhood. The kids going to schools or going for play in the playgrounds. In these places, mosquito bites the individuals, and till the time adult worms are alive, no symptoms are usually seen except for some transient infections of like we will have some sort of thread which is like infectious, which will be reddish in color. We will find some fever happening there. Other than that, there is no infection in that initial part of the disease. But in the later part of the disease, we will have acute attacks. In the acute attacks, what happens? The fever is very high, and one can see the lymphatic channel which has been infected. The whole area becomes really reddish and high in temperature. And eventually, when the adult worms die and the disease gets into the chronic form, in that specific phase, we find the swelling, which I just discussed in the previous slide, and which you can see on the right side as well. And this swelling accompanies with the thickening of the skin, and we also have the problem if the genitalia are engaged, especially in the males. We have hydrocele as a problem. There is a huge accumulation of fluid there. In addition to that, if somebody's kidneys are engaged, there is chyluria. There is a severe protein content coming out with the urine, which is milky white urine. People sometimes regard this as a problem, which is something sexual disease or something like that, but it is mostly filaria, and there is no cure as I discussed earlier. Next, please. So, in terms of the burden of disease, if we see globally, there are around 1.4 billion people who are at risk living in those geographies. There are 72 countries total where LF has been an endemic problem. And we do have some numbers, but these numbers are tip of the iceberg because everywhere the reporting of the cases is not that accurate. If we see India also has around 0.74 billion. In a way, like half of the population of India is living in LF endemic areas, and this contributes to more than 50% of the global burden. That is one of the biggest reasons why we want to work on lymphatic filariasis and try to finish off the disease as soon as possible. We have around 20 states and UTs where the disease is prevalent. There are 228 districts where the problem is there, and the numbers I will not discuss here. Next, please. So if we see the map of India, the ones which you find in yellow are the areas where we have no infection, or perhaps we do not have data to say that these are LF endemic areas. But the ones in the green are the areas where lymphatic filariasis has been found. And out of the total number of districts, we have 133 districts where the microfilaria rate is less than 1. And since that is the target we have to achieve, these districts do not conduct any mass drug administration campaign. Rest of the areas do the campaign, and I will speak about that in the subsequent slides. Before going to that, just to keep everybody informed, we have two specific pillars for elimination target, which is by 2030. So one pillar is to reduce the infection to a level which is less than 1% microfilaria or less than 2% of antigenemia level. And the second pillar is largely to support the people who are suffering from the disease, that is, people with hydrocele and lymphedema. So if both these services are provided in any country, then only that country will be eligible for elimination validation. Next, please. So, in terms of the strategy, what WHO has designed, that if we find an LF endemic district, there will be 5 to 6 mass drug administration rounds, and these areas are known as administrative units. In our local paradigms, we call them in the work program side. It's an implementation unit, and after these 5 to 6 rounds with high coverage, more than say 85% of the beneficiaries covered, we will stop the MDA and we will conduct a transmission assessment survey. So we have a pre-task and one is a task, in which what happens, the individuals are checked using filaria testing strips. And if all the people who are living in that area who are checked, then if the total number of people are less than 2%, then we say the transmission has been interrupted. And after 2 years, we again do a transmission assessment survey, or followed by a third test after 2 years. If all the tests are passed in that specific unit, we call that this area has reached the elimination target. If any of the transmission assessment areas are failed, then that geography has to undergo master administration at least 2 rounds so that they are eligible for pre-tests and tests subsequently. When we do the surveillance of the disease, we have like two mechanisms. One mechanism is known as annual surveillance. In India, we have each individual implementation unit has four sentinel sites and four random sites. So currently, what happens? Night blood surveys are being done in those geographies, and these four sites are like three in rural areas and one in urban area. So what happens in these areas? The team goes over there and they have to collect sample of at least 500 people in that specific geography. So nearly everybody is eligible for this testing. The problem what we have in the night blood slide testing is that the microfilaria comes in the body during the night time, mostly after 10 p.m. till 2 a.m. And now getting people, are ready to take the samples in the villages during that night time is a real challenge. And even the community is not very, cooperative if you go at, 11 o'clock or 12 o'clock in the night for sampling. So this is one of the big, reasons what we have, and that is why we are talking about the new LF diagnostic, mechanism. So night bed slide is one mechanism. The second is availability of filaria testing strips. So once a specific area reaches to a level where the microfilaria is less than 1%, then they are eligible for the pre-test and test. And in pre-test and test, we use the filaria testing strip. This is a strip which is having blood samples, and there is a lateral flow system through which we can figure out whether antigenemia is there or not. Now, the challenge what we have in the use of filaria testing strip is that a person who has been infected has taken the treatment and all the microfilarias have been killed, but still the person will be antigenemic for a long time duration. So the person may not be transmitting the disease, but the person will be antigenemia positive. So getting to know whether the infection is currently there or not is a big challenge using filaria testing strips. We will discuss more about this in the subsequent slides. Next, please. So globally, there is a platform known as Global Program for Elimination of LF, and they had signed up for elimination of LF across the globe long back in 2000. And the aim was to finish off the stuff in 2020. But we found that by 2020, not all countries reached that target, and now the targets have been revised for 2030. Next, please. Now, if we see the government data which is available at National Vector-Borne Disease Control Program Directorate, we find that per year, most of the time, there was a coverage more than 90%. Now, this is a government admin reported data, and we have some challenges because when these districts are checked for transmission assessment surveys, many of them actually failed tests. So the message what we have is that even if the coverage is shown as high, but if the people have not taking the drugs, not consuming the drugs, perhaps the transmission cannot be interrupted. So we do not have strong tools as of now through which we can tell whether people have consumed the drugs or not. But when we go ahead and if we have strong diagnostic tools, we can actually connect the dots between coverage as well as the transmission of infection. Next, please. As of now, only 133 districts out of the total 328 districts have reached to a level where they have interrupted the transmission. And if you see over last like 9-10 years, the stagnation of achievement of success has been there. Like we have not been able to reduce the transmission, and the blue bar is actually still at the same level. So we are aiming to increase the coverage of drug as well as reducing the transmission. That is the government's plan, and they are doing a lot of things around that. Next, please. This is a map which shows the success, like those districts which have passed TAS 3. You can see in dark blue color, mostly in the southern states of India. And then you see TAS 2 and TAS 1 also are mostly in the lower part of the country. But the districts, the states like UP, Bihar, Jharkhand, West Bengal, Chhattisgarh, Odisha, we are still having challenges over there, and the mass drug administration campaign is being continued there. Next, please. In terms of the infectivity and the markers which we have, so when the person becomes infected, the first and the foremost marker which comes out is the L3, which is in mosquitoes, and the L3 comes into the human body. And then in human body, we have antifilaria antibody which comes up, followed by circulatory filarial antigen. And then later on, by 6 to 9 months, microfilarias are also visible in the blood. Next, please. Now, as of now, what is being done is, as I told you, is the night blood slide. So the thick blood smear is taken to detect the live microfilaria in the slide, and then test to detect Wucheria bancrofti circulatory antigen is there, and there is a company which is coming up with LRA name, and Abbott is the manufacturer which has this particular testing strip. There have been some efforts on ELISA, but they have not been optimally utilized in the community because of multiple challenges. We will discuss that. Then for Brugia, we have only antibody test as of date. We do not have any antigen based test. PCR techniques have been tried, but they are not very widely used across the country, and there are key challenges we will discuss in the subsequent slide. So, what we are trying to speak about is the current options what are available for diagnosis are very limited. Let's go to the next slide, please. So this is slightly a busy slide, but let me take you through one by one. So if you see the signal on the left side, we have parasite in the vector. Now, if we have a vector, the indicator will be L3 larva in the vector or filarial DNA. Now, if you want to diagnose, we can dissect the mosquito and we can see whether the L3 larva is visible or not. But if you want to do RT-PCR, it is not a easy stuff. We have not tried in the program as of date. And the comments what we have is that the measure of transmission potential is there, but it is not widely used. So if you see the limitations on the right side, the dissection techniques are very challenging, and people in the field do not have those type of skill sets. So an RT-PCR requires laboratory capacity and specialized equipments. So genome monitoring is not practical for all the programs. Although Government of India is signing up for strengthening their genome monitoring system, but as of date, we do not have that widely used. For filarial DNA, the challenges are that PCR requires lab capacity and specialized equipment not available in all the areas. If we talk about the human beings, in humans, we can have microfilaria in the blood sphere, and this is a direct measure of infection, which is very specific. The problem for MF is that if we take the children less than say 10 years of age, those children do not have many microfilarias in their bloodstream. They may be antigenemia positive. But for microfilaria, we all understand which requires microscopy. We have to have good technicians who will look into it. And going at night time to do this is a very challenging thing which we cannot do every time. The second option for humans is like having circulatory filarial antigen, and we have the lateral flow assays which I referred in the past. We do have ELISA options not used in the program. So on antigens, what happens is that this measures very well about the Burgeri bancrofti. It strongly correlates with the microfilaria, but more sensitive than microfilaria detection. It's highly specific, and rapid tests are available. So we do have it, but the amount of FTS produced globally is very less. It's only one company which produces this FTS kit, and we will be seeing that how much India will be requiring moving forward. Now, the challenge for the filaria antigen test is the presence does not confirm viability of adult worm to reproduce. Like even if the adult worm is there, we do not know whether the production of microfilaria is happening or not. For that, we will have to go for the night blood slide. So, no antigen test available for Brugia, which I discussed. In certain settings where the triple drug regime of ivermectin, DC, and albendazole is used for MDA, circulatory antigen alone may not be sufficient for making decision to stop the MDAs. So that is one of the important reasons why we want to discuss with all of you regarding new diagnostic tests. Then we also have filarial DNA, which can be used through PCR, but again it requires a huge lab and the capacity in India is not that robust. And then we have indirect host. We will have antibodies in the human beings. Again, for this, we have lateral flow and testing for antibody testing only for Brugia, not for Burchiria. ELISA is not done in the program. Now, the challenges for the antibody test is it is difficult to distinguish recent exposure from historic exposure or infections. So we will not be able to decide when to stop the like MDAs in those areas. Next slide, please. So, for more information, WHO headquarter has come up with a very nice document, and this has all the details about what is required for the program, what are the key challenges we have the program in the program. So, I will request all of you to please look into this particular document, go to this link, and try to download this document for further information. Next, please. Now, India is moving ahead from a district level implementation unit to a block level implementation unit. When we go and do this type of activity, the amount of slides collected and the amount of tests required for the country will be huge. We will not discuss this in detail, but this is something which is going to happen very soon in the country. Next, please. Now let's talk about the requirement of FTS for the country. As of date, like till last year, India only required roughly 2 or 2.5 lakh FTS kits to do transmission assessment surveys because the evaluation units were really huge, around 2 million. Now the evaluation units are becoming per 5 lakh. So the amount of FTS required has gone hugely up. So in 2022, the way we calculated, like Government of India calculated, it was 1.67 million tests required. That is, around 17 lakh tests required per year. And because the geography is huge, every year districts will have to conduct these pre-tests and tests. So at least for the next 4 to 5 years, if you see, the amount required is close to 20 lakh kits per year, and the company which produces the kits in US as of date can only produce maximum 3 to 4 lakhs for India, and for globally again 3 to 4 lakhs. So their maximum capacity is 7 to 8 lakh kits per year, test per year, sorry. Now if we want to do in India some sort of deeper dive, we will require so many filaria testing strips. So if you see at the bottom side, the requirement is really huge, and the market will be still there till 2030, because all the areas which will be eliminating the disease will have to showcase using FTS and the transmission assessment survey. One more thing I want to emphasize here is that it is not only India which will require these tests. There are around 52 countries which are doing MDS currently, and they will also be requiring this type of transmission assessment survey test using filaria testing strips. So the whole launch which we have jointly done with the Department of Biotechnology is basically to help India, and at the same time we are also looking forward to the global market. If India succeeds in developing good testing strips, and if they are validated by WHO headquarters, those tests will be required not only in India but in other countries as well. So this is about the scope and the market. And I think this is my last slide. Can we go to the slide, please? So yeah, this is the launch what Dr. Shishantu has just discussed, and we will be discussing this in detail. So next, please. Thank you so much. So from my side, I am done. So Roshetu, back to you, please. Thanks.
Thank you, Dr. Bhupendran, for a very informative presentation. We would now like to take you to the NTD call details and how to, submit a successful grant application for the program. So, as already mentioned by our colleagues earlier, NTD are known to affect poorest and marginalized population of the world from tropical and subtropical regions that particularly lack healthcare settings. Considering that India accounts for overwhelming majority of NTD cases, there is an urgent need for more sensitive and specific diagnostic to support community diagnosis and post-intervention surveillance of LF. Thus, aligning with Government of India's commitment to end neglected tropical diseases like lymphatic filariasis, GCI BIOMARKERS has announced an funding opportunity on diagnostic for lymphatic filariasis. This is GCI's seventh call for proposal. The call has been announced on 7 June 2022 for a period of 45 days with the proposal submission deadline of 21st July 2022. The call is basically aimed at harnessing the indigenous strength of biotech community of the country. Next slide, please. So the main aim of the call is to address an urgent need for the development of novel diagnostics for LF. The program will basically support diagnostic development in alignment with WHO target product profile that is intended for field use in national LF elimination program, primarily India and globally. The goal of the challenge is to have a reasonably inexpensive, durable, and accurate point of care testing method that can be used in developing and remote geographies. The successful diagnostic or novel approaches developed through this call would then be supported for further validation and deployment in national programs for LF elimination. Next slide. The diagnostic being explored for development under this grant would mainly include improvised version of existing test for more reliable mapping and post MDA stopping decisions, as narrated earlier by Dr. Bhupender. We may be also supporting alternative tests that meet or exceed the current filarial test strips or microfilaria detection tool. We would also like to support entirely new methods for identifying viable adult filarial worms in the human body, or entirely new methods specific for early exposure or pre-patent infection of ELF causative species. We are seeking improved biomarkers for stopping triple therapy MDA, need specific markers of worm viability to better assess impact of MDA or IDA or triple therapy on transmission potential. There may be biomarkers for improved post-MDA and post-validation surveillance, biomarkers specific for early exposure to confirm elimination of transmission and or to detect early occurrence are also needed. Yeah. So we are seeking proposals that provide a strong rationale for the work proposed, demonstrating a clear understanding of India's context and need, and present a defined hypothesis and associated plan for how the idea would be tested or validated. The program is designed or structured basically in two phases, that is phase 1 and phase 2. Currently, we are seeking proposals for phase 1 of the program. So basically in phase 1, there are two categories of project approaches in which proposals are solicited, and both hold equal value and importance. Under short-term approaches, the projects will be supported for a period of 9 to 12 months, depending upon the stage of development or available preliminary data. We will encourage projects which already have their proof of concept, prototypes, or target product profile ready for further validation or clinical testing based on their technology readiness level, that is TRL 2 to 6. Under long-term approaches, we will support studies that are exploratory in nature, testing new approaches, ideas, or hypotheses, or those seeking to develop proof of concept and prototypes. These studies will be supported for a little longer duration, for a period of up to 18 months. So basically in phase 1, we will support 6 to 8 grantees. And the critical driver of success for phase 1 of the program is to have a prototype available for initial field-based testing within 9 to 18 months period. And a successful field-based test should require a minimum of technical expertise or training for their operation. So basically, through two phases of the program, we will support out-of-box ideas to POC projects or products that require field testing, piloting, and validation before deployment. Subsequently, we will also support large-scale validation, manufacturing, and commercialization of products through phase 2 of the grant. So BIOMARKERS GCI through this call is not only granting funding support, we will be also supporting grantees in other forms in terms of IP filing. If needed, applicant, investigators, and startups are also provided assistance in getting access to incubation spaces. The investigators receive access to technical and regulatory advisors along with network of market entry business development professionals to help refine the proposed solutions and take the ideas to the next stage of realization. The network of highly qualified individuals will work with grantees to provide them technical consulting and help overcome other hurdles at the earlier stage of technology development. Besides leveraging the mentorship resources, successful projects also have the opportunity to apply for a follow-on funding support through various other schemes of BIOMARKERS as well. And now I will hand it over to Dr. Madhvi to provide you the information how to submit a successful grant application for this program. Thank you.
Thank you, Dr. Rachel. Good afternoon, everyone. I'm Chandra Madhavi, working at Grant Challenges India, BIRAC. So I'll take you through how to apply and who can apply and what are the timelines for the program. So the objective of this Grant Challenges is to engage the world's most creative minds from across sessions, organizations, and geographies. So we support high-risk, high-rewarded innovations on the most difficult and pressing needs. See, this Grant Challenge is the one among them. See, the development of novel diagnostics for lymphatic filariasis disease. See, the challenge goal is to have a reasonably inexpensive, durable, accurate point of care testing methods that can be used in developing and remote geographies. Next slide, please. What makes for a successful grant application? See, the proposals must provide a strong rationale for the work they proposed, and demonstrating a clear understanding of India's context and needs, and present a defined hypothesis and associated plan for how an idea would be tested or validated. And the proposal's approach must align with the objectives of the current RFP. And also the objectives and activities and milestones must be well defined in the proposal. The first and foremost point, novelty and innovation. Does the proposal capture the enough novelty to address the discussed challenges is very important. And the second one is approach and methodology. Is the research plan or the objective and proposal schedule and the implementation methodology and work plan adequately detailed, and it should be realistic. Coming to has the applicant provided a clear metrics for monitoring the project progress, including the milestones and outputs and expected timelines, budget, etc. And have the resources required over the time frame been comprehensively mapped? For example, like technical management of the people, equipment, and outsourcing needs that need to be defined well in the proposal. Also, anticipated difficulties and risks that may be encountered during the project. So, have mitigation plans been considered in case of failure? That also need to be proposed during the proposal submission itself. Coming to the future deliverables and translational feasibility, the relevance and clarity of anticipated outcomes and deliverables to future implementation of the projects and commercialization is very important. Coming to the sustainability and adaptability of the system, does the proposal solution taken into account the complexity of the proposed geographic setting and context? And it should be cost effective, easy to operate, and accessibility is important. Coming to the organizational and investigator capability, the team composition covering key scientific and engineering challenges that this challenge is seeking to address. Like, is the applicant competent to ensure the effective conduct of the proposed work? And the applicant team must have a relevant capabilities and appropriate experience for the same. Has the applicant provided letters of support agreements with any third party they would like to engage with during the different stages of the product development? And the final one is the best value. So the solution must replace the dependability in terms of cost, ease to operate, and accessibility. Next slide, please. Like, who can apply? An individual researcher, innovator, entrepreneur, NGOs, foundations, not-for-profit organizations, community bodies can apply. Since Grand Challenges India is an Indian-led project, it is necessary that the grantee should be an Indian national, having Indian passport, preferably living in India till the project completion. And Indian companies applying as a grantees should have registered under the Companies Act in 1956 and must have 51% of the equity or shareholding by Indian nationals only. And there is no bar of age, qualification or experience for Indian innovators or researchers or entrepreneurs, but commitment for the development of the project till the end to reach the social impact is must. And coming to the individuals, also can apply, and they need to either get incubated to Indian incubation centers, or it need to produce a certificate from the host or affiliated institution for extending infrastructure or administrative support for the project once their proposal is recommended by the Scientific Advisory Committee. For applying the call, you need to visit BIRAC website. So click on the call for proposal and then current call tab in order to view the call detailed description. Further details on how to submit proposal would also be available on the BIRAC portal. If you are already registered user, then kindly enter your login details and submit the proposal. If you are a new user, your company, organization, is not listed in the drop down on the registration page, then you need to register your company or company. Once you register your company, then your organization will be activated within 24 hours after activation of the login ID. So please create at least 1 day prior before the registration. Next slide, please. These are the timelines for the proposal submission. The call was announced on 7th June and the call closes on 21st July. The award will be announced somewhere in the October 2022. Next slide. Thank you, everyone. Now the forum is open for the question and answers.
You can type your questions at the chat box.
Which are you speaking? You're on mute, I think.
So you can type your questions or I'll in the chat box if you have anybody, or you want to go through the call details and come back to us with your queries and questions. I will ask Dr. Richa and Dr. Madhvi to give their type in their email
So
ID.
are you speaking something? Please unmute yourself and then.
Are you able to hear me?
Yes, you're audible. Yes.
Okay.
So yeah. So if you want to get back to us in due course, but you can. I will ask my colleagues, Dr. Richa, Dr. Madhvi, and myself, to put their email IDs and contact details so you can get in touch with us. But remember, the call closes on 22nd of July. Am I right? 22nd of July.
21st.
Sorry, 21st of July. I stand corrected. So never wait till the last date. Please see that the your applications get submitted at least a day in advance before the last date, because if it crossed the date, it will be very difficult to accept the applications, because it's impossible to accept the applications. If you don't have any questions right now, you can go through the call RFPs and other things and come back to us. We are always available over email, and our landline phones are there in the site as well. We will be available for any questions, any queries, any hand-holding, anything you need regarding the application process. Bhupinder, you want to add something?
No, I think this is right. So the team is available. People can come back to all of us, and we will be happy to respond to them. And if there are multiple questions, we can also organize another webinar taking all the questions into account. So we are available.
We will be doing that as well. So that's that. Dr. Richa, Dr. Madhvi, any last points?
Just give us a few seconds. I'm checking with the IT. There are some questions in the chat box. There we were not able to visualize.
So actually people want to know the grant amount for this call. So we may support projects from the funding support of 1,000 USD up to 300,000 USD. That is 80 lakhs to up to 2 CR. So that is the funding support, well, depending on the stage of development, whether it's in phase 1, phase 2, or short-term approaches or long-term approaches. Depending on the scope of the work, the funding assistance will be granted subject to BIOMARKERS due diligence processes. Thank you. So there are a couple of questions in the chat box. We are getting it from the IT team. So if we could wait for another 2-3 minutes.
Sure. Because I can't see the chat box, so I thought...
there is a question for a project led by academia. Is there any requirement of company involvement?
Sir, you want to take up this question?
So, as such, it is not a requirement that a company should come in, but always it is nice to see how you take it to the next level. as an academia, you must have done a very excellent innovation. Of course, you can apply. But at the exit stage of the project, we would like to see how this innovation gets exploited and brought into the system. So for that, it will be nice to have a discussion with the company and to bring it on the level. But prima facie, it will, if you have a plan into it, it will be nice to build a 360-degree team to bring in the innovation. Because ultimately, the next round is to see how it gets scaled up to be in the next level. So that's what is our suggestion basically. But definitely you can apply independently as well.
And then next question is, how many partners can be there?
Multiple partners, there is no restriction, but each partner should have a role into the program. It is not just to increase the number, but each partner, two academic partners, one company, one partner, or two companies, one partner, whatever it is, but each one should have a proper role and a justified role into the program.
The next question is there any turnover limit for the applicant in the last 3 financial years for applying?
See, a startup can apply for this program as well, so we don't have any turnover limit per se. If you are a startup, you can also apply for it.
Do we need any prior experience of the topic or any research paper regarding the topic?
See, it is always good to understand the field and apply. So if you have not experience in this field, it will how do you justify fitting your application into this field? If you have a related experience also, you can definitely try that. But it is you need to understand the landscape very well before you for submission of the application. That will help you to defend the application. Bhupender, you want to add something onto this?
So I think you have said very well. So anyone who wants to apply, if their concept is, on diagnostics, and if diagnostics have been proven by them for any other disease which is, which is related to this or which may be related to this, I think that should be fine. But without any information and without any sort of, exposure in the past, perhaps it will be a challenge for them to go far. So it will be nice for them to connect and, get more information beforehand. Oh,
sure.
There is a question, is the application format available as PDF or Word document?
Richa, can you tell them what sort of application format? We have an online application system. So
online application format, yes, sir. So it's an online application format. You need to get yourself registered online on the BIOMARKERS portal, and then you can submit the application form online.
And whether DSIR certificate mandatory?
No, not at all. Not at all. It is not mandatory. You can apply without, but you should have a space to do the work. We can help you to get an incubation space as well. But if you need to have a space to do the work, but DSIR certificate is not mandatory.
So there is another question: is ethical approval should be processed to collect human urine blood samples before application is submitted?
Before application is submitted, no. But if you are doing sample collection, the necessary IRB approvals, not before the application submitted. If you are granted the project, when you start the work, necessary IRB ethical considerations needs to be done as per the Indian IEC guidelines. But not before the application is submitted. Once it is approved, and you should have a protocol or whatever it is, it needs to be really as per the ethical standards.
Any more questions, Devendra?
No, ma'am.
Okay, thank you very much. Now we are done with the questions. Over to you now.
Thank you. Thank you. I think we, as said earlier, myself, my colleague Dr. Bhupender, my colleagues Dr. Madhvi, Dr. Richa are always available to answer any queries over email, over landline. If you are visiting BIOMARKERS office, get a time slot done from us. We will be happy to handhold you. And the idea of BIOMARKERS Grand Challenges India and the foundation is to enable innovators to take advantage of this initiative. So we will be happy to handhold you, to guide you down the process anytime, but just fix up a time and do visit us. Or you can have a call with us. We can have a one-to-one call through a virtual platform as well if you have any queries. So please feel free to. But just a third, that please remember that don't wait for the last day to apply. Apply well in advance so that you are comfortable with the process. And we are always available for any discussion. Bhupinder, any last thoughts?
So I agree with you that people should start, talking to us wherever they get stuck. We will be, very, very happy to share thoughts, ideas, connect the dots as well. So we are really eager to look forward to good proposals, and we definitely want some out of box thinking as well. So nobody should restrict themselves with a standard thinking. If they have something totally, new, we are really happy to, discuss with you in detail. So that is
Sure.
the only.
And we will enable your innovation in the maximum limit, including space, including connects, including bringing in the regulatory guidance if you need. Everything can be done. So we will encourage you to take advantage of this. So once again, I thank you all for joining on behalf of the Gates Foundation, Grand Challenges India, BIORAC, Department of Biotechnology. We thank, and my colleagues, Dr. Richa, Dr. Madhuri, who are leading this program along with the foundation colleagues. We thank you all for joining this webinar and look forward to for interaction, innovation, this very, very strong unmet challenge this part of the world faces. Let us all bring out innovations which will not only address our problem, but also have solutions for the global community as well. Thank you. Namaskar.
Thank you. Namaskar.
Thank you, sir.